In the shadow of the double helix, the human genome that we have spent billions mapping and decoding, lies a second, more fluid, and arguably more influential genetic code. It does not reside in the nucleus of your cells, but in the dark anaerobic twist of your colon. For decades, science dismissed the trillions of bacteria living there as mere hitchhikers, helping us digest a bit of fiber here and there. But a seismic shift is occurring in longevity science right now. We are discovering that these microscopic tenants are not just passengers, they are an on-site pharmaceutical factory. And their most potent product is a class of compounds called indole metabolites. These molecules, forged from the amino acid tryptophan by specific gut bacteria, are emerging as the missing link between the microbiome and the aging process. They are the postbiotics that can reset the clock on cellular aging, regenerate brain tissue, and seal the gut against the chronic inflammation that defines getting old. This is not a story about eating more yogurt. This is the science of how the microbiome controls your health span and how you can hijack this system to engineer a younger biological age. To understand why indole metabolites are being hailed as a fountain of youth, we must first understand what aging actually is at a cellular level. Aging is not just the passage of time. It is the accumulation of damage. Scientists have categorized this damage into the hallmarks of aging. First, we have cellular senescence. These are cells that stop dividing but refuse to die, turning into zombie cells that spew toxic inflammatory chemicals known as SASP into the surrounding tissue. Second is chronic inflammation, or inflammaging. This is a low-grade, persistent immune activation that slowly degrades tissues, damages DNA, and exhausts stem cells. Then there is mitochondrial dysfunction, where the power plants of our cells lose efficiency, producing less energy and more toxic free radicals. Finally, we see a loss of proteostasis, which is the accumulation of misfolded, junk proteins, like the amyloid plaque seen in Alzheimer's. For years, we looked for the solution to these problems in human genetics. We thought if we could just tweak a human gene, we could stop senescence. But we were looking in the wrong place. Recent research reveals that the gut microbiome directly regulates every single one of these hallmarks. The gut is the primary source of chronic inflammation in the aging body. As we age, our intestinal barrier, a lining only one cell thick, begins to degrade. This leaky gut allows bacterial toxins like lipopolysaccharides to seep into the bloodstream. The immune system sees this and launches a forever war, resulting in systemic inflammation that accelerates aging in the heart, brain, and skin. This is where indole metabolites enter the stage. They are the peacekeepers. They are the signals that tell the gut lining to tighten up, the immune system to stand down, and the brain cells to regenerate. Indoles are not a single molecule, but a family of compounds produced when specific gut bacteria metabolize tryptophan. You might know tryptophan as the chemical in turkey that supposedly makes you sleepy, or as the precursor to serotonin. But in the gut, tryptophan faces a fork in the road. If it goes down the host pathway, it becomes serotonin, or kynurenine, which can be neurotoxic in excess. But if it goes down the microbial path, processed by the right bacteria, it transforms into indoles. Let's meet the key players in this chemical family. If there is a star of the anti-aging world right now, it is indole 3-propionic acid, or IPA. Unlike other metabolites that are quickly broken down by the liver, IPA accumulates in the blood and can cross the blood-brain barrier. It acts as a super antioxidant. IPA is a more potent scavenger of hydroxyl radicals than melatonin. It doesn't just neutralize free radicals. It does so without turning into a reactive, harmful compound itself. It also functions as an Alzheimer's shield. Studies have shown that IPA can prevent the accumulation of amyloid beta fibrils, blocking the gunk from forming in the brain. Furthermore, it acts as a mitochondrial guardian, helping maintain mitochondrial function to keep the lights on in your neurons as you age. Next is indole 3-acetic acid, or IAA. This is a master regulator of the immune system. It acts like a diplomat, specifically targeting a receptor on immune cells called the aryl hydrocarbon receptor, or AHR. When IAA binds to AHR, it triggers an anti-inflammatory response, specifically boosting the production of interleukin-22. IL-22 is critical for healing the gut lining. It tells the stem cells in the gut to divide and repair the barrier, effectively sealing the leak that causes aging-related inflammation. Then we have indole 3-lactic acid, or ILA. Often produced by Bifidobacterium and Lactobacillus species, ILA has been shown to interfere with the ability of pathogens to colonize the gut. It also plays a role in neurodevelopment and neuroplasticity, helping the brain rewire itself. Finally, there is indoleacrylic acid, or IA. Produced by Peptostreptococcus species, this metabolite promotes the production of mucin, the slime layer that coats our gut. This mucus is the first line of defense. Without it, bacteria touch our intestinal cells directly, triggering inflammation. So, how does a molecule made by a bacterium in your colon fix a neuron in your brain? The answer lies in that ancient signaling pathway I mentioned, the aryl hydrocarbon receptor, or AHR. The AHR is often called a sensor of the environment. It sits inside your cells, waiting for a signal. For a long time, we thought it only sensed toxins. But we now know its true evolutionary purpose is to sense the state of the microbiome. When indole metabolites bind to the AHR, they act as a key turning on a lock. This activation triggers a cascade of anti-aging gene expression. It causes the downregulation of NF-kappa B, the master switch for inflammation. Turning it off reduces the inflammaging fire. It leads to the upregulation of NRF2, which turns on the body's internal antioxidant defense system. And it causes the suppression of mTOR. The mTOR pathway is a major driver of aging. Inhibiting it mimics the life-extending effects of fasting. Recent research on Clostridium sporgens, a major IPA producer, showed that its metabolites suppress mTOR in immune cells, preventing them from becoming hyperinflammatory. However, the microbiome doesn't just produce good things, it can also produce pro-aging toxins. A recent breakthrough study identified a metabolite called phenylacetylglutamine, or PAGLN, which accumulates in the blood of older adults and those with heart disease. PAGLN actively promotes cellular senescence and DNA damage. This sets up a biological battleground: indoles versus PAGLN. Healthy aging is essentially a ratio. If your microbiome produces more indoles like IPA and IAA and less PAGLN, you maintain a youthful cellular profile. If the balance flips, often due to a loss of diversity and fiber, senescence accelerates. This brings us to the extinction event occurring inside us. If indoles are so great, why do we age? The problem is that the factories that produce them, the bacteria, shut down as we get older. Studies on centenarians, people living to over 100, reveal a startling fact. Their microbiomes often look remarkably similar to those of people 30 years younger. They have maintained the youthful diversity that most elderly people lose. In the average person, aging leads to a decline in key bacterial families, specifically Clostridiaceae and Peptostreptococcaceae, the primary producers of IPA and IA. This creates a vicious cycle. First, you lose the producers, meaning you lose the bacteria that make indoles. Second, you get gut leakiness. Without indoles to stimulate AHR and mucin, the gut barrier becomes permeable. Third, systemic inflammation sets in as toxins leak into the blood. And fourth, further dysbiosis occurs because inflammation changes the gut environment, making it even harder for the good bacteria to survive. To stop this, we have to intervene. We have to re-engineer the gut environment to favor the indole producers. You cannot simply go to a store and buy indole pills, at least not effective ones yet. The half-life of these molecules is short. They need to be produced continuously, in situ. This means you need to cultivate the right bacteria. The heavy lifter here is Clostridium sporogenes. It is the primary producer of IPA. Despite its scary-sounding genus, causing some to confuse it with C. diff or botulinum, Clostridium sporogenes is a mutualistic hero. It uses enzymes to perform the Stickland reaction, converting tryptophan into IPA. The challenge is that it is an obligate anaerobe, meaning it hates oxygen, and it forms spores, making it difficult to simply eat in a yogurt. It relies heavily on cross-feeding from other bacteria to survive. Then there is Peptostreptococcus russellii, a specialist in using mucin and tryptophan to create indoleacrylic acid. And we have the lactobacillus species like L. reuteri and L. plantarum. These are the accessible ones found in fermented foods that produce indole 3-lactic acid. They are excellent starters for shifting the gut ecosystem. So how do we boost these levels? It is not as simple as eating more turkey. In fact, eating high loads of protein without fiber can backfire. If tryptophan sits in the gut too long without carbohydrates, it can be converted into indole, the parent compound, which is then processed by the liver into indoxyl sulfate, a uremic toxin that damages the kidneys and blood vessels. We want IPA, not indoxyl sulfate. To get IPA, we need to steer the metabolism. The steering wheel is fiber. Specifically, we need a strategy combining tryptophan and resistant starch. This is the golden rule. Tryptophan is the fuel. Resistant starch is the catalyst. When you eat resistant starch, it bypasses digestion in the stomach and arrives in the colon. There, it feeds the microbiome. The fermentation of starch alters the pH and provides the energy needed for bacteria like C. sporogenes to thrive and convert tryptophan into IPA efficiently. A study found that dietary fiber supplementation specifically inhibits the production of the toxic indole pathway and favors the protective pathways. Without fiber, the tryptophan rots. With fiber, it ferments into medicine. So, what is the menu for youth? It includes cooked and cooled potatoes or rice. Cooking gelatinizes the starch, and cooling it for at least 12 hours, a process called retrogradation, turns it into type 3 resistant starch. This is rocket fuel for indole producers. Green bananas are another source, high in type 2 resistant starch. Oats and barley provide beta-glucans and resistant starch. And legumes like lentils and chickpeas are the perfect package of tryptophan precursors and fiber. Another strategy is polyphenol loading. Polyphenols, the color in plants, act as prebiotics. A recent clinical trial showed that a polyphenol-rich diet significantly increased serum IPA levels in older adults. Foods like pomegranate contain L-diltannins that boost Akkermansia and Gordonobacter, which support the indole network. Berries and dark chocolate are also potent sources. We can also use symbiotic stacking. Since taking C. sporogenes directly is difficult, we use a combination of probiotics and prebiotics. You are seeding the gut with lactobacillus, the infantry, to create the acidic environment that allows the native Clostridia, the special forces, to wake up and produce IPA. But you can eat all the resistant starch in the world, and if your lifestyle kills your microbiome, you won't produce indoles. We must consider sleep and circadian rhythm. The microbiome has a clock. It oscillates day and night. Poor sleep or shift work disrupts this rhythm, leading to a decrease in beneficial metabolites. Melatonin is structurally similar to indoles. Disruption in one often correlates with disruption in the other. Maintain a strict sleep window to allow the night shift bacteria to work. Exercise is also crucial. Cardiovascular exercise specifically increases the abundance of butyrate and indole-producing bacteria and increases the transport of tryptophan into the gut compartment. Zone 2 training has been linked to the highest microbial diversity. And finally, we need to look at fasting and gut rest. Constant grazing prevents the migrating motor complex, the gut's housekeeper, from sweeping away debris. A cleaning wave happens only when you haven't eaten for about 4 hours. Time-restricted feeding gives the microbiome time to switch metabolic modes, often enhancing the production of secondary metabolites like IPA. We are on the cusp of a revolution. Biotech companies are currently developing Indole Metabolites, drugs that mimic the shape of IPA to activate the AHR receptor without relying on bacteria. Startups like Propion Inc. are investigating nutritional formulas designed to block the enzymes that degrade tryptophan into toxins, forcing the pathway toward the rejuvenating IPA. However, we don't have to wait for the pharmaceutical version. The machinery is already inside us, it just needs the right raw materials. For centuries, we searched for the fountain of youth in mythical lands. It turns out, we were carrying it with us all along. The fountain is not a place, it is a process. It is the alchemical transformation of food into information by the trillions of organisms in our gut. Indole metabolites represent a paradigm shift in how we view aging. It is no longer just about the inevitable wear and tear of human cells. It is about the collapse of a partnership. By restoring this partnership, by feeding the indole producers, sealing the gut, and embracing the fiber-rich diet of our ancestors, we can unlock a powerful, innate system of cellular regeneration. The secret to staying young isn't in a capsule. It's in a cooled potato, a handful of berries, and the silent, invisible work of the bacteria that call you home.